Ready? Okay, perfect. Okay, well, good morning everyone. Just quickly before we start, I'd like to do a quick disclaimer. My presentation is probably slightly different to a lot of the presentations you've already seen. We're not going to be discussing my own research. Instead, I'm going to be talking about a few concepts and principles I've learned throughout my undergrad, and I'm hoping that you leave today with a little bit of a greater understanding of molecular biology, but most importantly, a bit of curiosity as well. Okay, so without further ado, the molecular basis of biotechnology. Cool. So everyone please close your eyes for a second and imagine a world where you can edit genes as easily as you can text on a Word document. Where you can program cells like you do on a computer, or where you can grow organs in laboratories. Open your eyes now. No need to imagine, okay? This is the world that we live in. And this is all thanks to the groundbreaking field of biotechnology. Today I'm going to take you through a journey of the molecular landscape that forms the foundations of modern biotechnology, and we'll explore how understanding of it has led to the most fundamental, and we'll explore how our understanding of life at its most fundamental level has led to revolutionary applications in medicine, agriculture, and environmental conservation. So we'll delve straight into the intricate world of DNA, RNA, and proteins, and the cellular machinery required for certain processes to unravel. We'll uncover how key historical discoveries paved the way for today's biotechnological advancements, and we'll explore the molecular and cellular principles that underpin these technologies. We'll also see how knowledge is applied to create solutions to some of humanity's greatest challenges. By the end of this talk, I hope you'll understand why I believe that biotechnology that is rooted in our ever-deepening understanding of molecular biology is not only shaping our present, but is also crafting our future. The story of modern biotechnology begins with James Watson and Francis Crick, their discovery of the DNA's double helix structure in 1953. This was built on Rosalind Franklin's X-ray crystallography work. That's the picture over there. This groundbreaking finding provided the first insight to the molecular basis of heredity, and in years that followed, scientists worked together to decipher the genetic code. A major breakthrough came in 1961 when, forgive my pronunciation, Marshall Nirenberg and Heinrich Matthei decoded the first word of the genetic code, showing that the sequence of 3 uracil bases coded for the amino acid phenylalanine. This marked the birth of molecular biology as we know it today. The field took another huge leap forward in 1972 when Paul Berg created the first recombinant DNA molecules from different organisms. This was then followed by Herbert Boyer and Stanley Cohen's creation of the first genetically modified organism in 1973, where foreign DNA was introduced into bacteria. These discoveries set the stage for a revolution in biotechnology, leading to numerous applications in medicine, agriculture, and industry. But at the heart of this preservation lies the central dogma of molecular biology. That is, essentially DNA makes RNA, which then makes proteins. This concept was first articulated by Francis Crick in 1958, and it is fundamental to our understanding of how genetic information flows through biological systems. This process begins with DNA replication, which ensures that genetic information is copied faithfully into the daughter generation. This is then followed by transcription, where an RNA copy of the gene sequence is created, and then translation, which turns that RNA copy, or uses that copy as a template to create a protein. Understanding these processes has allowed scientists to manipulate the flow of genetic information, essentially reprogramming cells for various biotechnological applications. A key tool in this manipulation includes the use of restriction enzymes, which can be referred to as molecular scissors, if you like. These essentially can cut DNA at very specific sequences. We call these sequences restriction sites. And they can be combined with DNA ligases, which are used to join DNA fragments together. This provides the basic tools we need for genetic engineering. The universality of the genetic code, with very few exceptions, means that genes from one organism can be expressed in another organism, which is a fundamental concept for creating genetically modified organisms. Recent developments, I'm sure all of you have heard of CRISPR, such as CRISPR. They have further revolutionized the field. This gene editing tool is actually derived from bacterial immune systems, allowing for incredible precise genome editing, opening up new possibilities in medicine and biotechnology. The applications of biotechnology are diverse and impactful, spanning fields from medicine to agriculture, environmental conservation. In medicine, biotechnology has allowed for an era of precise medicine. Monoclonal antibodies, for instance, are engineered molecules that can restore, enhance, and mimic the immune system's attack on cancer cells. Gene therapy, which delivers functional genes to replace defective genes, offers potential cures for lots of genetic disorders. In agriculture, biotechnology has helped to address challenges like crop yield, pest resistance, and nutritional content. For example, genetically modified crops such as BT cotton, which you might have heard of, it actually produces its own insecticide, allowing for increased yields, and then also reduces the need for actual chemical insecticides. Biotechnology also offers innovative solutions to environmental challenges. So bioremediation, using microorganisms to clean up polluted sites, that sort of thing. Also in industry, engineered enzymes are making processes more efficient and environmentally friendly. Think of detergents and stuff. A lot of those detergents are actually using enzymes that we have extracted from cells. And this is also making industries a little bit greener. So a particularly useful mechanism for engineering enzymes and proteins, and one that I want to spend a little bit of time explaining, is called the PET expression system. PET stands for plasmid expression under the control of T7 RNA polymerase. Now, that sounds like a lot, but essentially all you need to know is that a plasmid is this little region of DNA here. These often occur naturally in bacteria, but we also harness these plasmids and engineer them in such a way that we can reinsert them back into a bacterial cell and then express genes that we would like to express. What we're looking at here is we've got the native E. coli genome over here with an included piece of included genes to that code for the T7 RNA polymerase. The T7 RNA polymerase has a really strong affinity for a T7 promoter. So when the bacterial genome is creating the T7 RNA polymerase, it then has a very strong affinity for the promoter, which is upstream of our gene of interest, so then we can express our gene of interest that's sitting in our plasmid here. Now remember, this is taking place in a cell, so all the cellular machinery that we require for this to happen is inside the cell. This expression system was developed by Studier and colleagues in the 80s, and then it was further developed by Novogen. So now there exists a wide variety of different PET derivatives that have different applications. This tightly controlled high-level protein expression system has become a very invaluable tool in biotechnology. It enables for large-scale protein production from various applications, including structural studies, enzyme engineering, and therapeutics. This PET system, along with earlier discoveries in genetic engineering, transformed biology from an observational science to one where where living systems can actively be manipulated, which is paving the way for advancements in areas such as genetically modified crops, gene therapy, and personalized medicine. Another key tool in biotechnology is the polymerase chain reaction, or PCR. Now, we all live in a post-COVID world, so I'm sure we've heard the term PCR a little bit here and there. But essentially all this does is it allows for the amplification of a specific DNA sequence. And this had applications in the Human Genome Project and things like that. So in order to faithfully explain the process, I'm going to try and tell this in the style of a story. Imagine if you found a very rare gemstone in a cave or something, and you are maybe not from the area, so you don't understand the lore around this gemstone, but it's very rare, so you want to replicate it. If we swap gemstone to DNA, essentially what we can do so if we pretend that the gemstone is actually DNA, this acts as like a template. And it's the starting material that we need to amplify our precious material. So think of it as, since you are not from the area and you don't know the lore, you're going to need some guides or someone to help you with it. We'll call those primers. So essentially, in the world of DNA, our primers will bind to the specific region of our DNA of interest, and then that will allow us to isolate the area and then amplify our specific gene of interest. Now the real magic will happen in the thermocycler, where a machine that can precisely control the temperature of your DNA samples. It's like a cauldron. You can, the temperature is raised and lowered in a careful kind of geography. So it's heated to a scorching temperature, causing the DNA to unzip, which allows for the single-stranded secrets to be revealed. And this is that denaturing step, where the DNA is stripped bare, ready for the next step, which the temperature is then lowered and allowing our primers to come in and bind to our complementary sequences of DNA, ready to guide our DNA polymerase, which is going to be our cloner, I guess, our replicator, to start making lots and lots of copies of this DNA of interest. So the real magic is actually that this is a continual cycle. So we'll cycle through the denaturing process, the annealing process, that's when the ends connect together, and then the extension process, over and over and over again. And with each cycle, the number of DNA copies doubles. So you go 1 to 2 to 4 to 8 to 16, blah, 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 blah. And you get a lot of copies of that DNA really quickly. What started as a tiny fragment now becomes a treasure trove of identical DNA sequences. These allow for study, analysis, and there's a myriad of applications available there. So at the start of the presentation, I asked you to close your eyes and imagine. You don't have to close your eyes this time. But the molecular revolution that began with the discovery of DNA structure has brought us to an extraordinary point in human history. We now have the ability to read, write, and edit the code of life itself. Now that we use this ability, how we use this ability will define not only the future of biotechnology, but the future of our species on this planet. Again, imagine a future where personalized medicine is the norm, with treatments tailored to your genetic profile. Crops are resilient to climate change, ensuring food security for billions, where we can reverse environmental change and preserve biodiversity, and where we have sustainable bio-based materials replacing environmentally harmful products. These aren't dreams, they are the potential realities that biotechnology is working towards. So, as we leave here today, I encourage you to stay curious, stay informed, and stay engaged with the evolving world of biotechnology. The molecules that we've discussed today may be microscopic, but their impact on our world is truly massive.